if I can introduce Tony Goldberg from the University of Wisconsin-Madison, which I certainly know has some climate extreme events in terms of between summer and winter in that campus. So without further ado, I'll introduce Tony to talk about drivers for emerging issues in animal and plant health.
Well, thank you very much. It's a pleasure to be here. Looking in the audience, I see that most of you are old enough to remember when nature used to be fun. And then you'll note that people like the speakers today have essentially ruined that for everybody. And we now know that the narrative has changed quite a bit over the last several decades, such that now our relationship with nature is a little bit less magical and a little bit more microbial. This is a typical drawing of how we perceive nature today, and you can see that bats and primates figure prominently, which is not surprising. And these sorts of realizations are one of the reasons why I focus my work on primates, and in particular primates in a national park in Uganda, in a biodiversity hotspot within this black area called the Albertine Rift, which has exceptional primate biodiversity and biomass and a very high rate of human population growth. And the interesting thing about working in this location for over 20 years is that we've watched the primates go extinct in the unprotected areas outside the park. And we know from more studies than I have a chance to document that a lot of this has to do with infectious disease. So we've asked people in this area simple questions about the relationship between disease and animals. So for example, simply, can people share diseases with animals? And as you can see in these three villages, most people say yes, some people say no, and some people simply don't know. One gentleman thought about it carefully and said, well, yes, I think so, because we're all coughing and so are the goats. If you want to know about, that's an interesting one. I'm not going to tell you what it is now, so you'll have to ask a question or find me at the coffee break, but that's an interesting story. But as we asked open-ended questions of the people in our study communities, we would get interesting stories like this. Yes, primates can bring diseases to people because we hear people telling us stories, like 2 years back or 3, that a red colobus, which is a kind of monkey, bit my 2 children and they died. We don't know if it's a poison from their teeth or a disease from their blood, but yes, they also have diseases. So we started to hear these anecdotes that sounded suspiciously like viruses. So before it was trendy, I have to say, we decided to go virus hunting in this population of primates by collecting some samples in collaboration with the Uganda Wildlife Authority and others. And we found lots of new viruses. Here's our evidence for 3 novel simian retroviruses, a simian immunodeficiency virus, a simian T-cell lymphotrophic virus, and a simian foamy-virus, all of which are very different genetically from anything that had been found before. I'm proud, I suppose, that more than half of the known relatives of simian hemorrhagic fever virus exist in the primates of Kibale. These are deadly Ebola-like viruses if you're a monkey. And just so it's not all bad, we found some good viruses. So we found the first example of an old world monkey PEGI virus. And in humans, this is a famous virus because if you're infected with HIV and also infected with human PEGI virus, it delays the progression of AIDS. So it's good. And in fact, we found that in the West African Ebola outbreak in the lower right there, that people infected with human PEGI virus, GBV-C, were less likely to die from Ebola infection. And we found a homologue of this virus in monkeys, and now we've developed it into an animal model to understand the pathogenesis of interaction between this virus and other viruses. So it's very exciting stuff. And yet, as I've been doing all this work in Uganda, and we've come up with this picture of the RNA viral diversity in this population of monkeys, I've begun to wonder. Every one of these dots is a virus, and every one of these dots is a story, and I could stand up here for hours and tell you about every single one of them. But I asked myself, given this knowledge, are we really any closer to predicting which of these viruses will cross into people? So and I love this type of work. I do it in my lab. I have my own deep sequencer. It's really fun. This is a really nice paper by Simon Anthony and his colleagues trying to estimate the number of unknown mammalian viruses in the world. Estimated here is 320,000, and fact, 2 years ago, it would only cost $6.3 billion to discover them all. Money well spent. But there is a school of thought that discovering pathogens for pandemic prevention is the way to go, that we should just simply discover them all, and in so doing, we will prevent them all. I think that's worthwhile, and I do this myself, but I think there's a complementary approach that we've been missing. I call it pathway discovery. Rather than focus on the pathogens themselves, let's also focus on the transmission pathways that they traverse. We should hunt, like we do for viruses, for unknown routes of pathogen transmission. And the analogy I like to make here is with the great age of exploration. In the time of Magellan, his mission was not to discover new continents and new islands, but rather to discover new trade routes or new pathways between known ports. And in discovering these pathways, these trade routes, he happened to discover new islands and new continents, but it wasn't the main goal. So what do I mean by a transmission pathway? Well, this is a simple way of looking at it. This is a forest edge habitat, and inside the forest live monkeys with their social network, and outside of the forest live people with their social network. And one of the goals of this approach is to identify critical nodes in the human and primate social network that contact each other for reasons having to do with their demography and behavior. And once we know the structure of these relationships, it should be possible to map the transmission of a pathogen through this network and out to the world. So I'd like to give a few examples of what I mean. This is a paper we published last year called Beyond Bushmeat, where we interviewed people in this area about their direct contact with primates. And here, this histogram shows the percentage of people who reported direct contact with each of these species of primate. And what you'll see by the modes of contact is that not a single one of them was bushmeat. There is no bushmeat in this area per se. These are all strange modes of interaction, like being chased and bitten by monkeys, finding monkeys dead, butchering them to give to the dogs, guarding your crops against monkeys, rogue chimps that come out of the forest and steal babies and bite people. Strange modes of transmission that are well beyond this paradigm of bushmeat that developed from the story of the origins of AIDS. And asking people specific knowledge about zoonotic disease, we found that their knowledge was really quite limited, and limited by sort of the common educational campaigns you'd see in the area. So for example, well, it's not showing up very well, but you can see that worms are commonly associated with pigs because of the known risk of trichinella. Birds, one gentleman did hear about bird flu, so that association was made, but this is a very incomplete knowledge base. We replicated this study in Nigeria with a student of mine, Sagan Friant, and this is in a completely different setting of a hunting community. So these are people who absolutely positively do hunt bushmeat for a living. And even here, the perceived risk, as you can see in the upper right, was much higher. 89% of the people acknowledged that there was a risk of zoonotic disease. But when asked what precautions they take against it, the answers were a little bit unsatisfying. On the lower right, you can see some of them just decided to cook the meat well, some avoided contact, some of them simply prayed. So knowledge about prevention is not complete as well. So in a sense, this shouldn't be surprising to anyone, but lack of education may be the most important of the common pathways. And simple lack of education about germ theory, hygiene, public health, and zoonoses that we see as rampant in a lot of these settings could be considered a pathway by which pathogens can enter the human population. I think we all know that. Here's a more concrete example. That's me on the left taking GPS readings on some cows. And as I was walking around this particular patch of forest, we discovered some ears of maize with a strange substance on them. That turned out to be a noxious combination of sand, hot pepper, and cattle dung. So why in the world would people put that on their ears of maize? It turns out they were doing that to deter crop raiding by this very annoying animal, the red-tailed monkey. But if you think about it, what a fantastic way to facilitate cross-species transmission of foodborne, fecally transmitted pathogens between cows and people who are going to eat that corn, and cows and monkeys who are also going to eat that corn. So it's a strange cultural practice that has the inadvertent effect of enhancing this type of transmission. And it's perhaps not surprising that we discovered the very first example of assemblage E Giardia duodenalis, which is typically a ruminant assemblage Giardia, in monkeys. Because in these environments, there is a lot of practices that are bringing monkeys and cattle into contact they never would have had before. And we also find human primate transmission cycles as well, which is not as surprising. This is a fun example, again, from Nigeria, coughing monkeys. So again, my student Sagan was very clever. She was working with a semi-captive group of red-capped mangabees, and she noticed a strange parasite egg in their feces. The parasite was Paragonomus, which is known to be transmitted through the ingestion of undercooked shellfish, specifically crabs. And we didn't know it until she found this, but it turns out the monkeys in this forest were eating land crabs and becoming infected with Paragonomus. So she did a clever experiment where she dewormed the monkeys, as you can see on the left, and was able to decrease their eggs per gram of feces, and at the same time she was able to cure their cough because they were coughing from this lungworm parasite. So she was able to prove that this was causing coughing in monkeys. What was interesting is that she reported interviews with local hunters. Hunters in this area report using primate skulls and feces to treat cough. In interviews, hunters made reference to seeing monkeys in the area cough as justification for these traditional remedies, indicating an intriguing link between parasitism, clinical disease, local beliefs, and primate conservation. So the idea here is that there are cultural practices that are actually born out of infectious diseases that have the inadvertent effect of perpetuating the transmission of those very same infectious diseases. So it's a strange circular feedback loop. This idea is not new, but it's been a little bit forgotten. It was first really talked about by Paul Ewald in the 1990s, and it's called cultural vectors. These are sets of cultural practices that together act like a mosquito to bring pathogens from other places to people without people having to move. So on the lower left, that's a classic example of waterborne zoonotic disease, where that set of practices, collecting water and washing clothes in a polluted urban riverway where animals also frequent, is a vector like a mosquito that can bring pathogens to you. So the examples I've given you, maize daubing and the coughing monkeys, are cultural vectors. And the interesting thing about these cultural vectors is that Ewald argues that they not only increase transmission, but they also select for pathogens that are more virulent, pathogens that don't care so much about laying low, but rather replicate to higher levels and can be more severe. I don't have time to go into the details of that argument, but it'll come up later. So what about vectors? Are there vectors as well that we might want to pay attention to? Well, this is a tick, and it's a nymphal tick of the genus Amblyoma. And what's surprising about this is not that it's a nymphal tick of the genus Amblyoma. It was where it was discovered. It was discovered inside my nose. So this is the famous Kibale forest nostril tick, which I happened to come back from the forest one day and felt a pain in my nose, and with the tweezers and a mirror and a flashlight was able to extract this specimen, and just out of curiosity sent it to a colleague of mine in Texas who's an expert in tick genetics, and we sequenced it and found that its genetic sequence did not correspond to any known species. So this was either a new species of tick or an old species that had never before been sequenced. And in case you may remember this, it went viral. I was all over the place in Science and National Geographic and all these full page spread in The Guardian. I was very proud of that. But why was there a tick in my nose? It's a very strange thing. Well, I asked my colleague Richard Wrangham, who's a famous primatologist, if I might go through his photographs of chimpanzees at my field site in Kibale National Park, because he had been doing a study of chimpanzee faces. So we went through the archived photographs, and looking at these photographs of baby chimpanzees, you can see that there are ticks in their noses. Over 25% of these chimps had ticks up their noses. So I had a chimpanzee tick in my nose, and I discovered it after I'd already flown back to the United States. Why would a tick go into the nose of a chimp? Well, if you're a chimp, then if you know any chimps, you would know that they're they pay a lot of attention to grooming. They love to groom each other all the time. So if you're a tick on a chimp and you're out in the open, so to speak, you will be groomed off, eaten, and killed. So you have to hide. And the nose is a wonderful place to hide because it's warm and wet and vascular. So if you can get there, it's great. So this is an example of a vector that thought I was a chimp, and who knows what pathogens might lie in these strange sorts of vectors that remain undiscovered. So my argument about drivers and public health is that really, in addition to discovering all the world's pathogens and classifying them, we should try to disrupt their pathways. Clearly targeted education, that's a no-brainer. But this is going to require a new approach. It's going to require mixing methods from the biological and social sciences. And it's going to require what I call disruptive technologies, technologies that can specifically disrupt transmission pathways. So here's an example, back to Uganda, of soil-transmitted helminths. We've done so many studies on worms in this setting, and here you can see two common soil-transmitted helminths, Trichuris and Esophagostomum. Trichuris the whipworm, Esophagostomum the nodule worm, and the clades, where I have a Z, are new clades where we've discovered evidence of zoonotic and reverse zoonotic transmission. In other words, the worms were genetically identical in people and non-human primates in the region. And we've also found just a ton of hookworm and cyclostoma and Necator on the right-hand side. So we know from this location that soil-transmitted helminths are a big problem, and there are many helminths transmitted through the soil. So we invented a pathway-disrupting technology. This was funded by the Gates Foundation. We know from soil-transmitted helminths like hookworm that many of them enter the feet, or that having soil on your feet increases the probability of transmission. And the solution is very clear. You need to wear shoes. The problem is that many people are not aware of the health benefits of wearing shoes in places where soil-transmitted helminths are endemic. So we looked around at our study area and noticed that people tended to like to wear shoes of this style. This is a typical slide-style sandal. And if you see on the top, there's this nice place where you might be able to put some information about the health benefits of wearing shoes. The problem is, what could you put there? Because literacy rates in this population are only about 10%. So you can't write, wear these shoes in order to prevent soil-transmitted helminths, because that assumes a lot of knowledge. So we used a holographic image, actually a lenticular image, as our new disrupting technology that flips back and forth using only pictures to show the benefits of wearing shoes to blocking the transmission of worms. And this project has been picked up by Tom's, the philanthropic shoe company with whom we're working right now, to see if we can adapt this technology for worldwide distribution across cultures. So this is just an example of a pathway disruptor that doesn't target a single pathogen, but rather recognizes an important common pathway that many pathogens share, targets that pathway, and tries to disrupt it to benefit people's health. So why focus on pathways? I think in the end, we really have to look to Mr. Darwin. If we focus on the pathogens, we'll find lots of interesting pathogens. But if we focus on the pathways and disrupt them, we can block the transmission of the pathogens we know about, we can block the transmission of the pathogens we haven't even discovered yet, and we can block the transmission of pathogens that have not yet even evolved. And if the concept of cultural vectors is true, then we can, in so doing, select for, in a sense, less aggressive pathogens, pathogens that want to lay low and not cause so much disease, or else they'll be disrupted, so we can foster the evolution of lower virulence in pathogens by focusing on blocking these common pathways. So I will stop there, except to acknowledge the many people who've helped with this work, and to apologize in advance if I've made any unwarranted leaps of faith. But I'll take any questions if there are any.
Thank you very much. There's time for quite a few questions here. What music to my ears is when you talk at the end there about the kind of focusing on the pathways. Is I know that if I was actually speaking to a minister it's a kind of great value for money when you can deal with several things at the same time rather than a single thing. So questions at the back there.
I've got a reaction to virtually your very last comment about the fact that if you block one pathway, you would slow evolution of those those infectious mechanisms. I have a feeling what you'd actually do is encourage evolution in totally new ways of transmission.
Great question. Yeah, so that's the part of this that I've glossed over a little bit. The question is, if you block these pathways, will you alter the evolution of the pathogens? And your point was that you might actually force them to find other pathways or other, and that's true. But if you think about it, if the argument goes something like this: if you are a pathogen and you are replicating to very high levels and harming your host, then you'll only be transmitted if there's some mechanism that actively brings you from your sick host to a new susceptible host. And the corollary is, if you block that mechanism, then you will favor the pathogens that replicate to lower levels, wait for longer, and are transmitted more slowly. So you're right, it's more complicated than I painted a picture of, but I think the important point is that we should focus not only on the transmission part, but we should be able to use evolution to our advantage. And if we're careful, try to, just as you say, push pathogens along an evolutionary trajectory. A colleague of mine makes the analogy, we can make influenza into the common cold the same way that we've domesticated wolves and made them into dogs. We could do that.
Time for another question, if there is one. Yep, front there.
Yes, the problem with blocking one pathway is you tend to come to over-rely on it. So like with drinking water in the 1990s, they over-relied on chlorination, and then they let the sand filters not work properly and cryptosporidium came through. So if you put in a block, let's say the shoes, then they may become over-dependent on those shoes, and that may let another pathogen through, I suppose is what I'm trying to say.
Sure. So this strategy is not, so the question is, if you block a pathway, won't you then become reliant for public health on blocking that pathway? And the answer is yes, but unfortunately, I'm not aware of any public health strategies for emerging infectious diseases that do not require sustained effort. So I think the challenge is to imagine creative pathway disrupting technologies that work more or less on their own and cheaply. I don't know if my particular silly example of the shoe is one, but I agree with you that if we're overly dependent on expensive technological fixes, we'll open the door to future outbreaks, and it won't be a good thing. So that's really an engineering and public health challenge we have to face.